morning. My name is Jose Puente, Senior Editor of the Pathogenesis, Virulence, and Health Response section of the Journal of Medical Microbiology. Welcome everyone to the fourth session of this year's seminar series of the Journal of Medical Microbiology, and thank you for joining us today. Just a few notes before we start. The live transcript is available if required. If you prefer to hide it, just click on the live transcript button and select hide subtitles. The session will be recorded and the link will be sent to you afterward. It will be available to view for 7 days. Please use the Q&A function if you have any questions you would like our speaker to answer during the Q&A time at the end. Feel free to use the chat to introduce yourselves and for general comments. Today is my great pleasure to introduce our speaker, Dr. Lysangela Alves. She's an editorial board member of the Journal of Medical Microbiology in the Medical Mycology section. Dr. Alves has a degree in biological sciences from the Federal University of Paraná, a master's degree in sciences in biochemistry from the Federal University of Paraná, and a PhD in cellular and molecular biology from the Osvaldo Cruz Foundation. She has been a public health researcher at the Osvaldo Cruz Foundation at the Carlos Chagas Institute since 2012. Her main area of research is molecular microbiology, addressing the transport of RNA molecules in extracellular vesicles and their role in pathogenesis and modulation of host gene expression during infection, using fungi, Cryptococcus neoformans, and Candida auris as models. Today, Dr. Alves will discuss the role of extracellular vesicles and antifungal response in Candida auris. Dr. Alves, thank you for being with us today. The floor is yours.
Thank you very much for the kind introduction. Let me share my screen. Is everything okay with the presentation?
Looks good.
Okay, thank you. So again, thank you for the opportunity of the Microbiology Society to have these seminars and the chance for us to present our work here, which is about the role of extracellular vesicles and antifungal response in Candida auris. So today my talk will cover some aspects of fungal infection, the antimicrobial resistance in fungi, Candida auris, the response to caspofungin, the extracellular vesicles, and the host response. So starting with fungal infection, it's been published in January this year, an update study where they showed that 6.5 million people are affected with fungal infections in the world, and 3.7 of these people die annually from fungal infections. It's the 5th death cause worldwide. Talking about Candida, 1.5 million develop invasive candidiasis, and almost 1 million people die from this invasive candidiasis. So these are impressive numbers, and they show how important it is to study these organisms. And when we talk about treatment, we have only 4 antifungal classes that are available for human treatment. One of them are the classes of the azoles. So they target the synthesis of ergosterol, so they block the lanosterol demethylase, the ERG11, so you don't have ergosterol being produced, so you alter the composition of the plasma membrane. The other class are the polyenes that also target the plasma membrane, and it acts by sequestering the ergosterol that is in the plasma membrane, creating pores. And one example of this is the amphotericin B antifungal drug. The other class are the echinocandins, and they act in the cell wall of the fungi, and it blocks the beta-glucan synthesis by targeting the FKS1 protein. So you alter the composition of the cell wall, and therefore you lead the cell to death. And the 4th class are the nucleoside analogs that affect the DNA metabolism. However, we are seeing throughout the decades an increase in antifungal resistance in different types, species of fungi. So in this study, they analyzed throughout the decades from 1977 to 2017, and we can see an increase in the species that have some kind of antifungal resistance. And this can be observed in plants, so in agriculture, and also in the human context. And the question is, why is antimicrobial resistance increasing? We have a lot of information about antimicrobial resistance in the bacteria context, and there's a lot of efforts, a lot of studies, and a lot of actions being done in this sense to study and try to comprehend better the antimicrobial resistance in bacteria and how to prevent this. And in the fungi, we don't have much of information, but what we can speculate is that the climate change is affecting the population, the fungi population in the world. The overuse of antifungals in agriculture that is increasing every year is also leading to antifungal resistance, especially it's being studied in Aspergillus fumigatus or other Aspergillus species, where they see this cross reaction of the azoles used in agriculture and how they are affecting the azole use in the hospital settings. As I showed you before, we have limited antifungal drugs available for human treatment, and we are also observing a change in the population profile. So we are living more, and so we have more people that are prone to develop antifungal infections. So this is in the context of the One Health that we need to think about and consider ourselves in the planet. So we are affecting the environment, and these are leading more resistant pathogens being identified, and they are emerging or adapting. And this affects the people in the hospital settings, and even can affect people that are not immunosuppressed and the health population also. So when we talk about resistance mechanisms in fungi, they are a little bit different from what we see in bacteria, but we can have a lot of different mechanisms that are occurring. So for example, for the azoles, we can have mutations we can observe mutations in the ERG11 gene. So therefore you block the interaction between the azole and the ERG11 protein, and you have a normal plasma membrane synthesis. Continuing the azoles, we observed polyploidy, so we increase the expression of ERG11, therefore you dilute the antifungal in the system. We also observe mutations in the efflux pumps, so we increase the overexpress the efflux pumps, so they secrete this antifungal out of the cell. And for echinocandins, we also can observe mutations in the FKS1 gene. So you block the binding of echinocandin to FKS1 protein, and you have a normal cell wall synthesis. And for amphotericin B, we have mutations in the ERG6, so you alter the composition of the esterol that is being introduced in the plasma membrane. So amphotericin B does not recognize anymore this esterol, or even unknown mechanisms that prevents by amphotericin B by sequestering the ergosterol that is in the membrane. So these are some of the mechanisms, and there are a lot of unknown mechanisms also that are being discovered every year. So taking all of that into consideration, that fungi, they are increasing in the anti-resistance, the mortality, the morbidity, the spread, so the distribution in the world. In 2022, the WHO published the first fungal priority pathogens list. in the same way that they've done for bacteria. So now they are addressing this for the fungi. And they divided different species in critical group, high level group, and medium group in order to have more people studying and trying to develop new drugs and new methods for detection and from different species. And the critical group involves Cryptococcus neoformans, Aspergillus fumigatus, and Candida albicans, and also Candida auris. Candida auris was firstly described in Japan in 2009 in the ear canal of a patient in a hospital. After that, it's been spreading in more than 50 countries in the 5 continents. We have cases and outbreaks of Candida auris. Most of the isolates are drug or multi-drug resistant, and I will come back to that a little bit later. It shows resilience in diverse environments, especially, and this is really alarming, in the hospital settings. It causes outbreaks in healthcare settings, and the routine identification is still difficult. Looking at the phylogenetic tree of Candida auris, we can see that in this branch, where we have Candida auris and also Candida emuloni that is close related to Candida auris, they are all human pathogenic species. They are all haploid and they are all related to multi-drug resistance. However, we can see that in Candida krusei and Candida glabrata, we have also a multi-drug resistance profile. So this is not only restricted to this branch of the Candida genus. The Candida auris is divided in clades. So first, it was 4 clades identified, being clade 4 and clade 1, the one that have more isolates that are being identified, but we also have clade 2. The Japanese first strain isolate was identified as clade 2 and clade 3. Initially, the clades were divided by geographic region, but now we see that, for instance, in the case of United States, you have representative from all the clades. So now people are using more the number of the clades instead of clades from South Africa or Asia and so on. And we can see here the distribution of these clades. It is believed, or because of genome sequencing, that they arose almost simultaneously in different regions of the globe. Recently, clade 5 was identified in Iran in 2018, and clade 6 was identified last year in Singapore and Bangladesh. I forgot to mention these clades, they can vary from 100 to 1,000 SNPs when you compare the clades, but inside the clades, the difference is not that great. So they are more similar. That's why we can group them in different clades. The characteristics of C. auris, it's a yeast, so it divides by budding, can grow individually or in clumps, depending on the isolate. The genome is haploid, as I told you before. The shape is ovoid, ellipsoidal, or elongated. It varies from 2.5 to 5 micrometers in diameter, does not form germ tubes as Candida albicans, rarely forms hyphae or pseudohyphae. This is observed under stress or specific conditions. It grows well at room temperature and grows up to 42 degrees. And the precise identification is done only by mass spectrometry. Other types of identification that is used in clinic sometimes misidentify Candida auris with other Candida species. And what else do we know about Candida auris? So it's the global mortality rate is approximately 40%, the crude mortality rate. This depends on the clade that we are talking about. So the range, it's from 30 to 75% of mortality. There are some predisposing risk factors for invasive disease. So people that are in ICU, that that have been exposed to antifungal treatments, immunosuppressed, the one that are under invasive procedures, ventilators, and catheters also. As I told you before, one of the profiles that is characteristic of Candida auris is the multi-drug resistance profile. So 93% of the isolates are resistant to fluconazole, 35% resistant to amphotericin B, and 7% are resistant to echinocandin. And 41% of the isolates are resistant to 2 drugs, and 4% of the isolates are resistant to all the antifungal drugs. And for these ones, we don't have treatment. And here we are looking at numbers, at crude numbers, but this is really serious. So to tackle that, I brought an information about a patient. So this is a study case from a 29-year-old woman from New Jersey, United States, that had short bowel syndrome. So on day 1, this patient underwent a multivisceral transplant, so liver, pancreas, small bowel, and colon. And she was receiving a couple of antibiotics, and also the antifungal caspofungin was on day 1 when she went to surgery. On day 4, they identified the first isolate of Candida auris. They sequenced the genome, and they saw that this initial isolate was already resistant to azoles by the ERG11 mutations, and also mutations in the efflux pumps that I show you in the beginning of the presentation. After a couple of days, this person was already, they identified other isolates from C. auris from this person. And in this moment on day 24, after 24 days being treated with caspofungin, when they recovered this isolate, they sequenced this isolate was already resistant to echinocandins. So here they identified the mutation of FKS1. So at this moment, they started to treat with amphotericin B, and they changed the echinocandin from caspofungin to micafungin. This person had a lot of problems. After some days, she started to improve in quality, and they continued to isolate Candida auris. And this led to use of other antifungal treatments as voriconazole, flucytosine, ibrexafungerp. And on day 69, they isolated more, they recovered more isolates, and this isolate here, the 2032, was PAN-resistant. So it was resistant to azoles, it was resistant to echinocandin, it was resistant to flucytosine, and it was resistant to amphotericin B, despite of the fact that they didn't sequence any mutation, but it was resistant because this treatment here on day 30 to 40 didn't work. And in the end, on day 74, the patient passed away. So this is only one example of how serious and how threatening Candida auris can be, and other fungi species that are developing antifungal resistance. So we need to study these organisms in order to get a better view of how they acquire these mutations, and how we could manage this in the hospital scenario. Candida auris also have a lot of virulence factors. So as I told you, it's highly thermotolerant, so it can grow at 42 degrees. It's highly osmotolerant, so high salt concentrations, this species can adapt very well. It's highly stress resistant, so in the abiotic environment, so in the medical devices, or even in fabric, it can stay up to 21 days. It produces a lot of hydrolytic enzymes and efflux pumps, expresses a lot of adenosines, and can form biofilm. And most of these proteins and molecules that are involved in these virulence factors, they can be present in extracellular vesicles. So we also can consider the extracellular vesicles as carriers of virulence factors in the fungi. So what are extracellular vesicles? So they are bilipid bilayer structures that are released virtually by all types of cells. So here I brought you an example of a gram-negative bacteria, a fungal cell, and a mammalian cell, where we can have these structures being released. They can have different origins and different names. So the International Society of Extracellular Vesicles, they recognize the use of the general term extracellular vesicles to term all of this structure. But they can have different origins. For instance, the exosomes, they vary from 50 to 150 nanometers in size, and they are formed via the endocytic pathway. So the multivesicular bodies are formed and released to the extracellular space. We can have a blabbing directly from the plasma membrane, and the name of these structures are microvesicles or ectosomes, and they vary from 100 to 400 nanometers in size. And the apoptotic bodies, as the name says, is formed during apoptosis, and they are over 1 micrometer in size. These structures, they are very interesting because they carry different molecules. They can carry proteins, they can carry different types of RNA, they can carry lipids, pigments. So many molecules can be carried, and they are involved in cell communication. And this cell communication can be intraspecies, so between fungal and fungal, and interspecies also. In fungi, there are a lot of studies addressing the fungal EVs and the host response. So I brought you some examples. So in this case, for Cryptococcus neoformans, the extracellular vesicles can modulate macrophages by inducing the increase of nitric oxide, increasing cytokine production, increase phagocytosis, and increase killing. For Paracoccidioides brasiliensis, another fungi, the extracellular vesicles, they induce the polarization of macrophages into the M1 profile, increasing the inflammatory cytokines. And for Candida albicans, for dendritic cells, the EVs increase antigen presentation and cytokine production. For the macrophage, similar to Cryptococcus neoformans, increase the nitric oxide production, cytokines, and NF-kappa beta activation. And when we use Galeria melonella as an infection model, and you pre-treat Galeria with EVs from Candida albicans, and after that you challenge with the yeast cells, the EVs protected Galeria melonella against Candida auris infection. So these are some examples of how the interaction between the extracellular vesicles and the host cells can be modulated. So the topic of the talk is Candida auris and antifungal response. So caspofungin is the drug used in therapy for C. auris as recognized by CDC. So we wanted to perform a comparative transcriptome and proteome analysis in the presence and absence of caspofungin, examining the yeast cell and also the EV content. So starting with the cell, we selected 2 strains of Candida auris, one that was more resistant to caspofungin and one that was more sensitive. We used sub-inhibitory concentrations because we wanted to see the growth during 24 hours and how the Candida auris would adapt to in the presence of the stress caused by caspofungin. After that, we performed some measures. So one of those was the ROS production, so reactive oxygen species. And we see that after 24 hours of treatment, we increased the ROS production in the 2 strains. We observe also the morphology of the cells under treatment. So here is the control. we can see the cells growing as individual yeast cells. And after the treatment, they grow as aggregates and large clumps. This has been described for Candida albicans also and Saccharomyces cerevisiae under caspofungin treatment. We also measured and analyzed the increase of manoproteins in the cell wall. So this cell wall from Candida species, they are formed by a chitin layer right up the plasma membrane, followed by a glucan layer, and finally the manoprotein layer. And as we don't have the glucan being correctly synthesized by the blocking of FKS1 protein, the cells can adapt and trying to change the composition, increasing or the manoproteins or chitin. In the case of Candida albicans, we observe that chitin is the prevalent molecule that are used during caspofungin treatment. But in Candida auris, we observed that mannans were more predominant. So here we have the cells under control conditions, here under caspofungin treatments and the two strains. And we can observe that we have a stronger signal when we have caspofungin treatment. We also measure this by flow cytometry, and we can see this increase, especially for this strain. We have a lot of manoproteins. So this is used to compensate the lack of glucan in the cell wall and allow the cell to survive. We also analyzed the transcriptome response in the presence of this drug. So we saw a change in many transcripts. So this, I brought only the upregulated transcripts in the presence of caspofungin, and they were related in gene ontology terms with nucleotide and sugar metabolism, ribosome, cell cycle, many types of N-glycan biosynthesis, fructose and mannose metabolism, and protein processing in the ER. In addition, we also observe a lot of cell wall related transcripts that were upregulated during the treatment. In accordance to the previous slide, the mannan metabolism, the transcripts were highly overexpressed. GPI anchor transcripts that code proteins, glucan metabolism, transcription factors related to cell wall metabolism, and chitin. In addition, we went to look at the virulence factors. So there are a lot of virulence factors already characterized in Candida auris, and we look at the expressions of these transcripts, and they are all upregulated during caspofungin treatment. And they are involved in different aspects of the cell metabolism. Adenosines and efflux pumps, we observe increase, morphogenesis and biofilm formation, lytic enzymes, and the stress-resistant kinase protein. And they are all upregulated during Casper treatment. We also analyze the proteomic response. So for this, we compare the control and the Casper treatment, and very similar to what we observe for the transcriptomic, we observe a lot of ribosomal proteins, a lot of proteins related to glucan metabolism, mannan metabolism that were upregulated in the cell. And when we compare the mRNAs in blue and the protein content in green, we see a strong correlation between the gene ontology terms that were upregulated in both. For example, the cell wall biosynthesis and degradation, GPI anchor, ribosomal protein, indicating that we observe a robust response of Candida auris during this 24 hours in the caspofungin treatment. And what about the EVs? We also studied this. So the first thing we did was to look at the microscope to see the morphology of the EVs. So this is how the EVs look like under electron microscopy. These are the controls. Here we included a third strain to do this analysis. And here is under caspofungin treatment. So we did not observe any change in the shape and the morphology of the EVs. we observe a slight increase in size in the particles. This is through nanoparticle tracking analysis. So they increase a little bit in size, but the interesting result we got is the particle concentration. So the amount of EVs being produced and released by the cells under treatment. So for these strains, for instance, we got almost 10 times more extracellular vesicles being produced under the Caspa treatment. And this is very interesting. And then we went further to characterize these EVs. So there were many molecules that were altered after the caspofungin treatment. One of them were the no-coding RNAs that are the most abundant molecules that we identify in the EVs. So most of the RNA molecules that we sequence in EVs, they are smaller than 200 nucleotides. And this is very in accordance to what we observe for mammalian EVs, that they have more microRNAs and small no-coding RNAs. And we also observe that for fungi. And among the no-coding RNA molecules that we identified, the tRNA was the most abundant one. So we identify in at least 2 strains and the 3 strains that we analyzed. And when we talk about tRNAs, we are not sequencing the full length tRNA, it's the tRNA fragment. And these tRNA fragments, they are being described as components of EVs in all types of cells. So in bacteria EVs, in mammalian cells, in cancer cells, in healthy cells, and fungi cells and parasites, they all secrete, they are contain these tRNA fragments. And here I brought you 2 examples. So for this tRNA for glycine, here is the full length tRNA, and in light purple is the region that we sequence in our data, showing that we have an enrichment in this case for the 3' region of the tRNA. And this one for tyrosine, again, we have the full length tRNA, and this is the region that we sequence. And it's not well known why these fragments are being directed to the EVs and what is the role in cell-cell communication. But this is a very interesting data. We also sequence antisense RNA, so the Candida auris doesn't seem to possess the machinery to do RNAi or microRNA pathway, but we did observe some antisense RNA. So here is the transcript, and here is the region when we have this short sequence alignment. And this is representative of the triplicate, but we observe this in the triplicate, and we observe this for the 3 strains that we studied. And the transcripts that the gene ontology related to these antisense transcript targets are related to biosynthesis of cofactors, protein processing, DR, spliceosome, DNA replication, mismatch repair, and mitophagy. We also identify messenger RNAs in EVs. They are not very abundant, but they are present there. So here, an example of the sequence coverage. So this is the transcript and the orientation of the transcript in the genome. And this here are the coverage of the transcript, showing that we have actually full-length messenger RNA in the EVs also. And then we compare in the 3 strains that we analyzed, we can have some transcripts that are isolate specific, like this, but we also have a core that is common from the 3 strains. And this 87 here, the gene ontology terms related to those are translation, cell wall, nucleosome, and extracellular vesicles. And this result is similar or in accordance to what we observe for the cell. We also identify proteins in the EVs, and it's interesting because the MDR1, the multidrug resistant protein 1, and the multidrug protein CDR1, they were identified as upregulated, so they are over-identified in the EVs during caspofungin treatment, and they are efflux pumps. And we also identify other types of proteins, ribosome, cell surface and cell wall, and extracellular vesicle. And we also perform some metabolomic analysis. And for this, we use caspofungin, the powder that we use to treat the cells, only the media that was prepared just like we prepare the EVs for the analysis, and the control, and the caspofungin. And here it's the example of only one strain that I brought you here in triplicate. So this is steroid and steroid derivatives. They were identified only under caspofungin treatment. So these 4 clusters here, we are analyzing it further, but it's very interesting because we didn't identify them in any replicate of the control, showing that we have proteins, we have messenger RNAs, and we have like lipid-like molecules that are being directed to the EVs under specific treatments, in this case, the antifungal caspofungin. And now the last part of my talk will be about the EVs and the host response. We have shown previously that when you treat the epithelial cells with EVs from Candida auris, we increase the capacity of the Candida auris to interact, to adhere to the epithelial cells. So here we measure the yeast cells and epithelial cells ratio, and we can observe this increase here when you pre-treat the epithelial cells with EVs, and after you challenge the epithelial cells with the Candida auris cells. Here we can see at the microscopy also this. So without EVs, you have in blue the epithelial cells and in red the fungi cells. And with EV, we increase a lot the number of adhered cells to the epithelium. And this was not observed for Candida albicans. And another interesting fact is we have some specific isolate response, because here we have 2 different isolates from Candida auris, and they display different profiles of interaction or adhesion. So to go further to this experiment, we wanted to see another type of cell, endothelial cell, and how it would react in the fungi-EV interaction. So we used C. auris, the EVs under control conditions, and the EVs under caspofungin treatments, the same way we did all the previous experiments, so subinhibitory concentrations, 24 hours. After that, we recovered the EVs and we treated the UVEC cells with these EVs. After 3 hours of interaction, we recovered the UVEC cells, isolated the RNA for sequencing, and performed some more adhesion experiments. So for the RNA-seq, here we have the UVEC responding to the EVs under control conditions. And we can see that there is a change in gene expression of the UVEC cells. So here we have 516 transcripts being differentially expressed, 277 were downregulated in the presence of the EVs, and 239 were upregulated. And when we look at the caspofungin, we have 171 transcripts being differentially expressed. 458 were downregulated, and 413 were upregulated during caspofungin in the EVs that were pre-treated with caspofungin in the UVEC cells. And we can see that the EVs, they modulate the gene expression, and then we compared. So this is the EVs from control conditions, and these are the EVs from caspofungin treatment. And we see that 568 transcripts from UVEC cells are exclusive, modulated when we have caspo in Candida auris cells, and 213 exclusive for the control, and 303 that were common to both conditions. And from these exclusive ones in the caspofungin treatment scenario, we have some downregulated messenger RNAs. And among those, I'd like to call your attention for the Notch signaling pathway that was downregulated. The Notch signaling pathway is involved in many processes in the cell to confer homeostasis on the cell, but it also plays a role during different infections, different pathogens. So for instance, it has a role during viral infection, bacterial infection, fungal infection, and parasitic infection. And looking closely to the fungal infection scenario, the Notch signaling pathway, it plays a role in different organisms. I brought two examples here. So for Paracoccidioides brasiliensis, the Notch signaling pathway acts by modulating cytokine production, IL-6, that are important in immune response against Paracoccidioides brasiliensis. And for Cryptococcus neoformans, mice deficient for Notch signaling in T cells, they have uncontrolled fungal infection. So they have a high fungal burden and decrease the rate of the mice survival, indicating that this pathway is important to fight fungal infection. And only the EVs from Candida auris, they were sufficient to downregulate this pathway, maybe to favor the infection. And last, we did some experiments about endothelial cells and fungi interaction. So similar to those that we did in the beginning. So here I have only the cell, UVEC cells and C. auris, and we calculated the ratio. Here we have UVEC cells pre-treated with EVs, control EVs, let's say, and Candida auris, and we have an increase in the adhesion in the interaction, similar to what we observe for the epithelial cell. And then when we treat the cells with the Caspo EVs, and then we challenge the cells to interact with UVEC cells, we have a much higher increase in the ratio of the interaction or the adhesion between the C. auris and the Caspo EVs, showing that the C. auris Caspo EVs increase the yeast cell adhesion by the composition of these EVs probably. So in summary, I hope I showed you before today that caspofungin caused morphological changes, aggregation of yeast cells, and modifications in the cell wall composition of C. auris. The EV quantity, protein, metabolite, and RNA content changed drastically upon caspofungin treatment. And there are differences between Candida auris strains, so we need to pay attention to that when studying Candida auris. We cannot assume a lot of conclusions driven by studying one strain. So we need to pay attention to that and study more strains to have a broader view of what is happening. And this EV composition seems to be regulated because it's not random. We have some direction of components that are being directed or enriched. The EVs modulate the endothelial cells gene expression, as we did by RNA-seq. EV increased the adhesion of yeast cells to the host cells, and the EVs are important means for cellular communication. So I'd like to thank all the people involved in this work. So this is my group inside the Gene Expression Regulation Laboratory. So I'd like to thank Bruno, Mariana, Caio, Isadora, Ricardo, and Luisi, because they did most of the work that I showed you today. I'd like to thank the other collaborations from our lab, Márcio, Flávio, and Haroldo, the collaborators at the Federal University of Paraná, Guilherme Sasaki, that he's performing some cell wall composition analysis for us, Dr. Leonardo Nimrichter from Universidade Federal do Rio de Janeiro, Dr. Taísia Fio from Unicamp that did the metabolite analysis for us, Dr. Craig Ennis and Clarissa Nobil from the University of California, and Drs. Joshua Nosanchuk and Daniel Zivnich Miranda from the Einstein College of Medicine, where I started to work with Candida auris. And the financial agency, CNPq, Fiocruz, and Fundação Araucária also. And finally, just to show you a little bit about Fiocruz. So Fiocruz started in Rio de Janeiro. The unit, the headquarters is there, and they have this very, very beautiful palace, the Moorish Palace, that was built in the 900s. And we have different units spread all over the country. And we are here, right below São Paulo. So this is the south region of Brazil. And this is our unit. We are about 300 people. And this is the city, Curitiba. So I invite you all to one day visit us because it's a very nice place. So thank you very much.
Thank you, Dr. Alves, for a really interesting talk. Dr. Matty, would you like to start with questions?
Sure. Absolutely. Thank you, Dr. Alves. It was a very nice talk, and I really enjoyed the contents, and I learned a lot of new things, and I was very happy to see all the fantastic work you're doing. And of course, I want to visit Brazil again. I was in Foz de la Iguazu, and it was amazing.
It's here, it's our state.
I know, and we should talk after the session ends about some of the initiatives you're interested in. Okay, coming back to there's a couple of things that really stood out for me was that the microvesicles, right? And you said the non-coding RNA, the RNA component is always they're fragments. Do you ever get a full length, or you always get a fragment?
Never sequence the full length. It's always fragments, and is not. And we don't have sequences in the middle, or we have the 3' end or the 5' end. So it's not random fragments of tRNA. They are directed. They are very specific.
They're very specific. It's very interesting. And how about in the human EVs?
Yes, a lot of.
Also,
yes, in the same pattern.
Okay.
Fragments of tRNAs enriched in the 5' or 3'.
So, I mean, this is shows my ignorance about Candida. So Candida has an entire translational machinery, or does it on its own? So is there any way to differentiate in the microvesicles if it's grown during infection, microvesicles that are host generated versus Candida generated during infection?
I think we can, because if we have ergosterol in the membrane of the EVs, and we don't have ergosterol in the host cell, because the protein content, we were doing some analysis on that. The protein content between the fungi EVs and mammalian cells EVs, they are very similar. So we have a lot of elongation factors, a lot of enolase and metabolic proteins that are conserved in eukaryotic, in different eukaryotes. So sometimes it's difficult to identify, but based on the ergosterol composition, we could definitely.
Okay. I just want to tell Dr. Puente, I have tons of questions, so interrupt me whenever. So the other interesting thing was talking about the vesicles, right? You said the exo, extro, and apoptosis vesicles. And can you, I mean, I'm asking experimental questions. When you extract the vesicles, so we have protocol to get the vesicles, how do you sort them out into these 3 groups?
Okay. We just sort out the apoptotic bodies because we use filters. So after we centrifugate the media, and we ultracentrifuge, and then we filter on 400 nanometers pore. So we get everything below 400 nanometers or even 200 nanometers. We can have this option also. And so we eliminate the apoptotic bodies in this moment, but we cannot be sure that we have only exosomes or only microvesicles. So we have a mixed population of that.
Okay, very interesting. So good to know. So the other thing is that you said early on that if you if you can immunize the animals against these vesicles, you get protection.
Yes, this result is a little bit varies depending on the species. So for some species, you get protection. For other species, you favor the infection. So it depends on the model that you are studying. But they are trying to use EVs as vaccines, as for you to produce some antibodies against the pathogens using EVs. There are some actions on that sense.
Okay, interesting. So it could go both ways.
Yes.
But well, I'm sure they can microvesicles, can also be used to deliver antibodies if we organize the membrane accordingly. So the earlier, that one patient you saw this continuous change in SNPs, right? They simply getting, it almost reminded me of HIV progress. HIV virus constantly changes its sequence, and hence we don't have a vaccine for that. Do you know that at least in this particular case, that this or is it general for Candida auris? It has a very, very promiscuous DNA polymerase.
So there is a study addressing that to see if the repair machinery was somehow inducing some strains to be more prone to develop mutations, but they didn't see any correlation about that, because sometimes Candida auris isolates are resistant to antifungal without having any alterations in the genome. So are other mechanisms that are unknown mechanisms that are driving this resistance. That's not through mutations in the hotspots from the target genes.
Wow.
So they didn't see any correlation about that.
Okay.
There are some clades that they observe more mutations and more resistance. So for instance, clade 2 is not related to any outbreak so far at the moment. And clade 4 has more resistant profile. So that also depends on the clade. So we cannot assume everything based on one isolate or something like that.
Right. But I think what it tells me is that we need to do comparative genomics. We have to do whole genome comparison of this against the sensitive strain. Then we could potentially see beyond just SNPs that vary, right? So I'm sure somebody's working on it. Can I continue, Jose?
Well, let me ask address one of the attendees' questions. Dahid congratulates you, Lysangela, for a great talk, and is asking, can you please elaborate on EV's isolation methods from Hubeck cells? How much of isolation efficiency is required for sequencing?
The UVEC cells, we sequence the cell transcriptome, not the EVs. The EVs derived from the fungi. So we have two protocols to isolate EVs, can be from the liquid media and can be from the solid media. So it was published by our group. So Flavia, that is on the list of the people that I thank, she developed a method to isolate the EVs directly from the plate. And we recovered tons of EVs from that. And when I used the antifungal drug, I used liquid media to be sure that all the cells were getting the antifungal instead of in the plate. And the amount of EVs that we recover, it's 10 to the 11, 10 to the 10 in quantity. So it's a very nice quantity. And we
Okay.
use to treat the UVEC cells what we saw in the literature. We kept that, we use kind of MOI. So we use it 5 EVs per UVEC cell.
Okay.
Nice.
Thank you. Kelly, do you want to go ahead and then I'll follow with my questions? So,
Sure, sure. So this caspofungin treatment you have, and they go from what looks like a single cellular level to an aggregate, right?
Mhm.
Can you recover that phenotype back? Can you then remove the caspofungin, and then now they go back to being a single cells? Can you do you understand
That's
my
a very, yeah, that's a very interesting question. We haven't done that, but it's very interesting. I have no
Okay.
idea.
Okay, great. So the reason I ask you that question is based on my background on Pseudomonas and biofilm formation. We have the wild type that forms a very, very layers and layers of things, they don't clump, but then they undergo a mutation during in the patient's lungs. You can force it outside, but it undergoes mutations in the patient's lung, and then they form aggregates. The only way to recover back the phenotype is for second site mutations. So we knew from going from non-aggregate to aggregate, we knew we were able to map back the hard way what gene was doing that. And then to recover that, we also took a couple of years to realize that there was a separate mutation that caused them to revert back. And then over time, we have identified it's not one single mutation that allows them to revert, but multiple different mutations. Can any one of them can cause, and we don't know the selection. And I think perhaps one of my major contribution in that field was that to show the aggregation that now becomes very difficult to treat is really driven by a host PMNs. So we activate the polymorphonucleosides from healthy individuals, right? And then you treat the Pseudomonas with activated PMNs, you can force that mutation on Pseudomonas. Or you can treat them with the excess of H2O2, hydrogen peroxide, you can also force that mutation. So our argument is that the oxygen radicals in the lungs of these patients, what causes the mutations. So you did do hydrogen peroxide experiment, I thought that, right?
Yes, we did. Yes, no, it's very interesting. You can look at that because we thought that maybe as we change the composition of the cell walls, we have more mannans and chitin, and maybe this could be leading to aggregation, but they can definitely look at that. It's very, very nice.
Thank you.
Thank you.
Go ahead. Yeah. Go ahead, Aze.
yes, thank you, Kelly. It was a very exciting talk, Lysangela. These microvesicles are fascinating. And what temperature do you use to grow your fungi for all these experiments?
These ones we did at 30, but we do 30 or 35, 37, sorry, depending on the recovery, but these ones were under 30.
Yeah, I'm asking because I think it's interesting that it's thermotolerant candida. And I guess one of the symptoms of candida infection is fever. So thinking this thermotolerance as an adaptation, do you think virulence might be enhanced if you grow candida 40, 39, or 40, thinking
Yes,
that candida
yes,
might
yes,
go through this environment during infection?
yes, I definitely do, because when you reach this temperature, other pathogens starts to die, and then we have even a higher option of Candida auris to spread more. And there is a study that shows an increase in the virulence factors expression when you increase a little bit the temperature. I'm not sure about 42, but at 37 and 39, there's a study showing that they increase the virulence factors, the expression, and the adaptation of the cell because it's highly resilient. You can identify after you get a patient infected with Candida auris, you detect in almost any medical devices that you test, even in the ceiling and in the curtains in the ICU. So it's really crazy how it is resilient to abiotic stress or abiotic environments.
And what about MB's production at 40 or 42? Do you see an increase in MB's production? How about the content?
Yes, we didn't test that at this high level temperature. We did at 30, 37, and we only observed the growth capacity at 42 and 45. So in 45 didn't grow, but in 42 it grows slowly, but after 48 or 72 hours, you have a plate full of Candida auris at this temperature. But we could measure, this would be interesting to look at the EVs.
Yeah, because I was wondering about what you were saying about this difference between the treatment or not with the antifungi. And I was wondering if the content is regulated somehow, or is just randomly modified depending on what the fungi is expressing.
Yes, what is well known is that the EVs are very dynamic. So if you change the media, you change the composition. If you change the temperature, the conditions, and then they are very dynamic in response. So we cannot rule out the fact that some of the things that go to the EVs can go randomly, but most of the things we observe, they are directed. They have some kind of direction because sometimes not very abundant proteins end up in the EVs, and we have a lot of no-coding RNAs that are nuclear, and they are in the EVs. Why they are there? So small nuclear RNAs are there. So we believe that maybe it's a mix. Some are directed, some are random because they blabbed from the plasma membrane. We cannot be sure of that. Yeah.
So in terms of all these omics that you did to compare the resistant and the more sensitive strain, and with and without the treatment, I think I missed that part. Do you see a correlation in terms of what might be generating the resistance for the more resistant strain? In terms of what they are expressing during the treatment with the Caspo?
Okay, for the cell, yes, we did observe some. I didn't bring all the data, but yes, we did. So there are some specificities depending on the strain. So the most resistant strain, it changed a little bit the profile when you compare the more sensitive one. And we try to look at both to the common transcripts between the strains and the differences, and they have some difference, but nothing that call your attention and say, oh, that's why we are observing this more resistant phenotype. But neither of the strains that we work at, they had the mutation in the FKS1 gene. The FKS1 gene was the wild type.
Oh, another experiment that actually was very interesting. This experiment where you see adherence after pretreatment with the MBs. So when you did the control with Candida albicans, that was pretreating with Candida albicans microvesicles?
Yes.
And then for auris was with auris. Have you crossed the treatment in terms of now treating Candida albicans with the auris microvesicles?
Yes, no, we haven't. We are planning to do this in the next few weeks to see, and even getting the EVs from caspofungin and treat the Candida albicans, because Candida albicans is completely susceptible to caspofungin, to see if we observe any change in the profile or the response. We are doing that.
Or the appearance.
I don't have anything to bring.
That would be very interesting, yeah.
Mm.
But let me, there are a couple of questions, additional questions from the attendees. With these changes on treatment, does this affect its resistance to cleaning of the environment and making the organism persist in the environment? I'm not sure.
I understand the question.
I understand the question. This change in treatment, does this affect its resistance?
So basically, if I understand that question right, is if we keep changing the treatment and then there are new organisms appear, and then the existing cleaning methodologies or chemicals may not work. That's definitely possible, right?
In the terms of antifungal drugs or disinfectants?
They're talking about the surface cleaning, I bet.
I get
okay.
disinfectants, yeah.
Yes, the surface cleaning is really difficult about Candida auris also. So most of the hospital and clinical settings, they use ammonium quaternary to clean, and it doesn't work very well for Candida auris. So we need to use hypochlorite or peroxide to eliminate Candida auris from the clinical setting. So the classic disinfectant, the classic cleaning doesn't work for auris. So there's another procedure on that. So CDC made a lot of tests, and they have like a protocol for when you identify Candida auris in the ICU or.
Okay. Can I follow up with a question, Dr.?
Sure.
Yeah. So when two associated, the first question is just for clarification, and then I'll follow it up with a question. So what kind of infection is mostly seen in patients with auris?
It's bloodstream.
Bloodstream infections.
Yes, it is.
No lung, but it's just bloodstream.
Yes, because different from Candida albicans, that is part of, actually Candida auris is believed to be part of our microbiota also from the skin, but it's not like Candida albicans. So if you do experiments of infection, in vitro infection or animal infection, the fungal burden is not that high. So Candida albicans infects better than Candida auris. So, but when you get to the bloodstream and then you have like the sepsis going on, and then you cannot control. But doesn't affect that much.
Yeah. So then, what is the mode of entry then?
Sorry, what?
Mode of entry. How does auris enter the bloodstream?
So you can recover C. auris from our skin. So when you have an immunosuppressed patient with a catheter or any other invasive medical procedure, it enters and it goes from there.
So in other words, auris is hospital-acquired infections.
Yes, is a nosocomial infection.
Okay, so
it's related to outbreaks in hospitals. Healthy people, there's none case described at the moment of a healthy person that developed invasive candidias from Candida auris.
Okay, fantastic. So then the follow-up question is, and maybe you already answered just now, was that the auris really took a center stage for a while during COVID, because we had a lot of auris infections during COVID, and that too, not early, but like in the middle or late COVID era. Do you want to comment on that a little bit, Dr. Alves?
Yes. So what is believed is that during COVID, we had a lot of the quantity of people that went to ICUs were higher, and maybe the sanitary measures in the hospital staff may not be as adequate as they should. and then it got spread very easily. So in the first moments of the COVID-19, we didn't observe, but we observed a gradual increase during, as you said, and maybe sometimes we can have a problem with the Candida auris is the identification. So depending on the country, depending on the region you are, you don't identify properly. So sometimes you have Candida auris in the patients and you didn't identify it. So
yeah,
I
I
don't know if I
think
answered your question.
yeah. I sort of answered. That's fine. No, but I don't know any hospitals. But then I'm not a hospitalist. I've not heard, at least in my time, that auris is part list of bugs that hospital tracks, right? And they ought to be tracking auris, and they also ought to be tracking their resistance profile, because it is becoming extremely resistant. So if a patient comes down with an auris infection, they already know what is the circulating auris isolate in that particular hospital. And that's why we have stewardship in the hospitals to especially AMR stewardship in the hospital. So
Yes.
maybe there is a serious need to have serious discussions with the hospital stewardship management, and to including auris in their portfolio, right?
Yes, because auris, you need to notify the government agents when you have auris, when you identify. So it's a demand, and it was recognized by CDC, and I think most of the health agencies, they notify when you have Candida auris outbreak or cases.
So one last question, Dr. Puente.
Go ahead.
Pardon me. Okay. So we see auris in bloodstream, and patients are immunocompromised. I'm also curious is, a microbiome, that is fungal microbiome, is not that well studied. It's in a growing area, right? Do we know any microbes or other fungal species sort of always with each other? Like, are there a coexistence of auris with other fungal organisms or bacterial organisms? Do we know that?
No. What we know is that it's in the same site as Malassezia sympodialis in our skin.
Malacesias.
Yes, and Candida auris, and there is a tropism for the axillary region and groins, and so you can recover. And also, they are starting to recover from urine, also Candida auris.
Oh, really? So you can actually do a urine test, which is non-invasive.
Yes.
But if it is already in the urine, then it's already entered the bloodstream, right?
Yes, that's true.
So it's too late.
Yeah. And they start doing swabs in the axillary region and then the groins. And they believe that because of the pH and the temperature, that it refers to these sites.
Okay. Thanks. So the last question is that I thought that was the last one, earlier one. Did they ever recover auris in the lung during a COVID microbiome analysis? Do you know anything about that?
No, I've never seen any study on that.
Okay. Anyway, with that, I want to thank you for a fantastic presentation and discussion. And then I'll let Dr. Puente continue. And then I will catch up with you later, Dr.
Okay, thank you, Dr. Marti.
Yeah.
Just a couple last questions. One from the attendees. Do you think this test could be repeated for other pathogenic species such as Aspergillus? Like the sort of studies that you did.
Okay, could you repeat the question?
That if the same sort of experiments that you did with Candida can be repeated with Aspergillus, with other pathogenic species?
Definitely, yes, for sure. And we are
Yeah.
doing as some of these experiments for Cryptococcus also, because Marcio Rodrigues, that is in our lab, he's a specialist in Cryptococcus, so he's doing a lot of experiments in this sense also.
Okay. And I just have one last question. These experiments that you also did with the HUVEC cells to look at the adherence of the Candida strains, is it known a specific receptor on the cell membrane that Candida uses to adhere?
No, because we wanted to see if the caspofungin DEVs were changing the composition of the UVEC cells, also the membrane, maybe to change because it's facilitating the adhesion. But we didn't look at the protein composition in the UVEC cells. But would be very interesting to do that. So I don't know.
Is it known on other cells if there is a specific receptor?
I have no idea. I cannot answer that, sorry.
No worries. Well, I would also like to thank you for a wonderful talk. Just let me wrap up and well, just thanking everyone as well. I would like to thank you again for your excellent talk and insights, as well as the audience for attending. A reminder that a link to the recording will be emailed to you and will be available to view for 7 days. The next session of the JMM seminar series will take place on Friday, September 27, the same time, when Dr. Kendra Rambach from the Texas Tech University Health Sciences Center will be delivering a talk that will be announced later. Well, thanks again, Lysangela. It was very interesting and nice meeting you. And I hope to see you around the editorial board meetings at another time.
Thank you very much. Thank you.